welcome to a deep dive
Oh, okay.
into Actinobacteria.
Right.
So this one is really for all you medical microbiology enthusiasts out there.
Yeah.
We are going to be cracking open Bergey's Manual of Systemic Bacteriology today.
Sounds good.
And we're going to be focusing on
Okay.
the roadmap of the Phylum Actinobacteria.
Right.
So this is going to be your express lane
Yeah.
through this bacterial universe.
I like it.
We are going to be highlighting all of the families and orders that are relevant to you and your work,
Okay.
breaking down these phylogenetic classifications.
All right.
And we are going to be addressing
Yeah.
some of the controversies and uncertainties in this ever-evolving field.
Excellent.
So if you're ready to explore the intricate branches
Yeah.
of the Actinobacteria family tree and uncover a few surprises,
Sure.
buckle up.
Okay.
This is going to be a fascinating ride.
So in Bergey's manual,
Yeah.
they organize bacteria based on their evolutionary relationships,
Okay.
primarily using RNA gene analysis. It's been a revolutionary tool, but it's important to remember that relying solely on one gene
Yeah.
has limitations and has sparked debate in the field.
Right, because it's like trying to understand a person's entire personality based on their height.
Exactly.
You get some information, but definitely not the whole picture.
Right.
So how do we get a more complete picture of these bacterial relationships?
So ideally, a multi-gene approach would provide a more nuanced understanding.
Yeah.
So by comparing multiple genes,
Yeah.
we can either confirm or challenge
Okay.
the relationships suggested by rRNA analysis alone.
Right.
And this is especially important in a field as dynamic as microbiology.
It makes sense that we need multiple perspectives to
Yeah.
really understand these complex organisms.
Right.
So does Bergey's manual acknowledge these limitations?
Absolutely.
Okay.
The manual clearly states that its classifications are based on the type taxon principle,
Right.
meaning that changes to existing classifications only happen with strong phylogenetic evidence from various sources.
So it's like a constantly evolving puzzle
Exactly.
with new pieces being discovered and old assumptions being challenged.
Right.
So to give you a sense of the scale of this puzzle,
Okay.
the Phylum Actinobacteria is currently divided into 6 classes:
Okay.
Actinobacteria, Acidomicrobia, Choriobacteria, Nitroliruptoria, Rubrobacteria,
Yep.
and Thermoliophilia.
Wow.
I noticed some quotation marks around some of those names.
Right, so that's all about consistency
Okay.
across bacterial taxonomy.
Okay.
Subclasses like Acidomicrobidae and Choriobacteriae have been elevated to class level
Okay.
for a more unified system.
So like a major renovation project,
Yeah.
streamlining and updating the whole system.
Yeah, exactly.
But for us,
Yeah.
the medical microbiology folks,
Yeah.
I hear one class stands out.
Yes, you're right.
Okay.
The class Actinobacteria is our main focus.
Okay.
It houses the orders we encounter regularly in clinical settings.
All right. So let's dive into that class then.
Okay.
What are the major orders that we need to know about?
So within the class Actinobacteria,
Right.
we have several key orders starting with Actinomycetales.
Yep.
This order has undergone some significant changes,
Right.
now restricted to only one family, Actinomycetaceae.
So Actinomycetales has become quite streamlined.
Yes.
What should we know about this family?
Within Actinomycetaceae,
Okay.
there are 5 genera to remember: Actinomyces, Actinobaculum, Arcanobacterium, Mobiluncus, and Varibaculum.
Got it.
Each genus is important in its own right,
Yeah.
but the genus Actinomyces itself has some internal complexities.
What kind of complexities are we talking about?
Well, the classification of Actinomyces is a bit messy.
Okay.
Some species within the genus are actually more closely related to other genera
Well,
than they are to the type species Actinomyces bovis.
Huh.
This suggests that we might see reclassifications in the future
Okay.
as more research emerges.
it sounds like even within a seemingly simple order,
Right.
there's still so much to learn and discover.
Absolutely.
What's next on our Actinobacteria tour?
Next is the order Actinopolis
Okay.
a monogeneric order, meaning it has only one family, Actinopolis
Okay.
containing the genus Actinopolis.
Gotcha.
There's been a notable reclassification here as well.
Okay.
The species Actinopolis erequensis
Right.
was initially misclassified
Oh.
and now falls under Saccharomyces halophila.
Wow. It's fascinating how scientific understanding constantly evolves.
Yeah,
Even with something as seemingly fundamental as bacterial classification.
definitely.
What's next on our list?
Bifidobacterialis.
Okay.
This order's placement on the phylogenetic tree is still debated,
Okay.
but its importance in the human gut microbiome is undisputed.
Bifidobacterialis. That
Yeah.
rings a bell.
Yeah.
Especially for those of us who are fascinated by gut health.
Yeah.
Which family should we focus on within this order?
Bifidobacteriaceae.
Okay.
It houses 7 genera,
All right.
with Bifidobacterium and Gardnerella being the most familiar, especially in the context of human health.
Bifidobacterium.
Yeah,
The friendly bacteria we find in yogurt and probiotics.
that's right.
So Bifidobacterialis is a crucial order to understand,
Definitely.
even with its uncertain placement.
Yeah.
What comes after this?
Next, we have Catanulaspiralis,
Okay.
a deep, early branch within the class, Actinobacteria.
Got it.
It consists of 2 families,
Okay.
each with just 1 genus.
Okay.
Catanulaspiraceae with Catanulaspora,
All right.
and Actinospicaceae with Actinospica.
So a relatively small order.
Yeah.
But as we've learned,
Yeah,
size doesn't always reflect importance in the bacterial world.
that's true.
What's next?
Corinabacterialis.
Okay.
This order is thought to be closely related to both Actinospiralis and Pseudonocardialis,
Okay.
with all 3 sharing similarities in their cell wall structure.
Okay.
However, the exact nature of these relationships is still under investigation.
Always more to learn. What makes Corinabacterialis particularly relevant to medical microbiology?
It contains 6 families,
Okay.
some of which you'll encounter frequently in clinical settings.
Yeah.
These are Corynebacteriaceae, Deutziaceae, Mycobacteriaceae, Nocardiaceae, Signiliperaceae, and Succumberilaceae.
Got it.
Nocardiaceae is particularly interesting
Oh, why is that?
because it appears to be paraphyletic,
Okay.
meaning it might need reorganization based on newer research.
Reorganization on the bacterial family tree.
Exactly.
That sounds like a big task.
It can be.
What are the most significant general?
So within Corynebacteriaceae,
Yeah.
you have the large and complex genus Corynebacterium,
Okay.
which includes Corynebacterium diphtheria,
Right.
the causative agent of diphtheria.
Oh, well.
And within Mycobacteriaceae,
Yeah.
you have the genus Mycobacterium,
Oh, okay.
home to Mycobacterium tuberculosis,
Right.
responsible for tuberculosis.
Those are some serious pathogens.
Yes, they are.
I can see why this order is so important in the medical field.
Yeah.
You mentioned that both Corynebacterium and Mycobacterium are large and complex genera.
Yeah.
What makes them so challenging to classify?
Their sheer diversity
Okay.
and the difficulty in classifying them accurately based solely on rRNA analysis.
Okay.
Corynebacterium, for instance,
Yeah.
contains a vast number of species,
Right.
and they don't neatly cluster based on their rRNA sequences. It's a bit of a taxonomic puzzle.
A puzzle with serious health implications.
Definitely.
And what about Mycobacterium?
It's a similar story
Okay.
with over 140 species.
Wow.
Some slow-growing, some fast-growing.
Right.
They don't all fit neatly into distinct categories
Okay.
based on rRNA alone.
So are there any newcomers shaking up the classification of Corynebacterias?
There are
Okay, tell me about them.
genera like Amyclococcus, Hoyacella, and Tomatella
Got it.
have been added to this order,
Okay.
adding another layer of complexity to its classification.
It's a dynamic area of research.
It is a
So far, we've navigated Actinomycetales, Actinopolis, Bifidobacterialis, Catanulaspiralis, and Corynebacterialis.
Yes.
What's next in our deep dive into the class Actinobacteria?
We'll explore the enigmatic Franchialis.
Okay.
This order is shrouded in uncertainty,
What if-
ongoing debate about its phylogenetic relationships. Some analyses suggest the families within Franchialis might not form a cohesive group.
Oh.
Instead, they may exist as independent lineages.
It sounds like we're entering a more mysterious realm of the Echinobacteria world.
Yes.
What families are we dealing with here?
There are 6 families to consider.
Okay.
Franchiaceae, Acidothermaceae, Cryptosporangiaceae, Geodermatophylaceae, Nakamuralaceae, and Sporichthiaceae.
Right.
Franchiaceae is particularly noteworthy as it houses the genus Frankia.
Frankia sounds familiar.
Yeah.
Why is this genus important?
It's known for its unique symbiotic relationship with plants,
Okay.
helping them fix nitrogen,
All right.
an essential nutrient for plant growth.
That's fascinating.
Yeah.
But you mentioned the entire order is under scrutiny.
Yes.
Okay.
So conflicting phylogenetic analyses,
Okay.
meaning different methods of analyzing evolutionary relationships, have led to different conclusions about the validity of Franchiales as a unified order.
Okay.
Can be taxonomic evidence, which looks at chemical markers within the bacteria.
Yeah.
Hasn't provided a clear answer either.
So Franchialis is like that group of friends where you're not quite sure how they all ended up together.
Yeah, that's a good analogy.
Intriguing.
Yeah.
What's our next stop on this Actinobacteria journey?
Let's move on to glycomycetales.
Okay.
This order is intriguing because of its possible relationship with a genus from another order, Micromonospiralis.
Orders mingling with each other.
That's right.
This is getting interesting.
It is.
Tell me more.
So glycomycetales might be closely linked to the genus Actinocatinispora,
Okay.
which belongs to the order Micromonospiralis.
Got it.
This link is based on RNA analysis, and some chemotaxonomic and phenotypic features appear to support this connection.
What kind of evidence suggests this potential relationship?
So both glycomycetales and Actinocatinispora have similar cell wall structures,
Okay.
both containing mesodiaminopimelic acid and lysine.
Got it.
They also both produce chains of spores on aerial hyphae.
Oh.
However, there are differences too.
What sets them apart despite these similarities?
Their phospholipid types are different.
Okay.
And they have different dominant menaquinones,
Okay.
molecules involved in bacterial metabolism.
Oh, gotcha.
These differences highlight the complexity of classifying these organisms.
Like they're speaking different dialects of the same language.
Exactly.
So what are the main genera within glycomycetales?
There are 3 to remember.
Okay.
Glycomyces, Haloglycomyces, and Stachybrandtia.
All right.
Each has its own unique characteristics.
Yeah.
But their overall relationship to Actinocatinispora remains a fascinating puzzle.
It seems like every step we take on this journey uncovers another layer of complexity.
That's right.
What's up next?
Next, we have a newly proposed order, Geangelales.
Oh.
Closely related to glycomycetales.
New kids on the block.
Yes.
What prompted this new order to be proposed?
The discovery of the genus Haloactinopolispora,
Okay.
it turned out to be closely related to the genus Geangela,
Got it.
which was previously placed in a different family.
Okay.
Creating a new order was the best way to accommodate these closely related genera.
I love how bacterial classification is constantly adapting to new discoveries.
Yeah, it's a dynamic field.
What's next?
Let's talk about Kineosporiales.
All right.
This order's exact placement on the phylogenetic tree is unclear,
Okay.
with possible relationships to Actinomycetales, Bifidobacteriales, and Micrococcales.
So Kineosporiales playing the field,
Yeah,
trying to figure out where it belongs.
that's right.
What family does it house?
Just one Kineosporiation,
Yeah.
with 3 genera to remember: Kineosporia, Kineococcus, and Quadrisphaera.
Got it.
Each of these has its own interesting features,
Okay.
but the overall order's placement remains a mystery.
Kineosporiales,
Right.
the order with commitment issues.
Exactly.
We'll keep an eye on that one.
Okay.
What's up next?
Now we come to Micrococcales.
All right.
This order has been quite the hotbed of taxonomic activity,
Okay.
with families merging, splitting, and changing alliances.
Oh, drama. It is.
I'm ready for the bacterial gossip.
So the phylogeny of the families within this order is
Okay.
incredibly complex.
Right.
Imagine a family reunion with distant relatives you never knew you had,
Okay.
and everyone's trying to figure out who's related to whom.
Okay.
We see 3 major subclades.
3 major subclades.
Right.
Okay, let's break this down.
Okay.
What families belong to the first subclade?
The first subclade includes
Yeah.
Butenbergiaceae, Bugorialaceae, Celidomonadaceae, Promicromonospiraceae, Rarobacteriaceae, Ruiniaceae, and Sanguibacteriaceae. That's a lot of families.
It is.
What about the second subclade?
The second subclade
Yeah.
includes Dermacoccaceae, Dermatophalaceae, and Intrasporangiaceae.
Okay.
There is some debate about whether Intrasporangiaceae truly is a unified family
Okay.
or needs further revision.
Always the revisions.
That's right.
What about the final subclade?
The third subclade consists of the family Microbacteriaceae,
Okay.
and surprisingly, the order Bifidobacteriale?
Wait, an order hanging out with families in a different order?
That's right.
That's a plot twist.
It is.
And it highlights just how complex and intertwined bacterial relationships can be.
Exactly.
But remember, these subclade relationships are still being debated.
Yes.
Different analyses using different data sets and methods sometimes lead to different conclusions.
That's right.
So lots of uncertainty about the big picture in micrococcales.
Yes.
But is there one family that seems to be holding its ground?
You're right.
Okay.
The family Micrococcaceae
Yeah.
is a well-defined group within Micrococcales.
But I have a feeling there's more to this story.
Yeah.
Even within a stable family,
Yeah.
there has to be some drama.
There is.
So even within this seemingly stable family,
Yeah.
There's plenty to unpack.
There is.
For example, the genus Arthrobacter.
Yes.
One of the main characters in this family saga
Right.
is known for its paraphyletic nature.
That's right.
Paraphyletic nature.
Yeah.
Refresh my memory on what that means.
Essentially, it means that the members of Arthrobacter are scattered throughout the family tree.
Okay.
They are sometimes more closely related to members of other genera than to each other.
So Arthrobacter is like the rebellious teenager of the family,
Exactly.
defying easy categorization.
That's right.
And just a rebellious teenager.
Yeah.
It's caused quite a stir in the family dynamic.
It has.
There have been numerous attempts to classify Arthrobacter species
Right.
into more manageable groups
Yeah.
based on RNA similarities, chemotaxonomic features,
Yeah.
and even a combination of both.
Yeah.
We have RNA clusters, subclades, and groups,
Yeah.
all trying to make sense of this unruly genus.
Right.
Arthrobacter definitely keeps things interesting.
It does.
Are there any other notable members of Micrococci we should know about?
We have Micrococcus,
Okay.
the namesake of the family,
All right.
which is closely related to the genus Citricoccus.
Gotcha.
Then there's Cucuria,
Okay.
representing one of the earliest branches in the family.
Right.
And Nestorynconia, grouped with the genera Cinnamonas and Yaniella.
Okay.
And let's not forget Rothia,
Right.
which gained a new member when the Stomatococcus was reclassified into this genus.
It's a family full of interesting characters.
It is.
So Micrococci is the well-defined but complex family
Yes.
in the tumultuous order of Micrococcaceae.
That's right.
What other families should we know about in this order?
We have Butenbergiaceae,
Okay.
composed of the Butenbergia, Minimonas, Solana, and Serenobacter.
Okay.
Then there's Bogorialaceae, initially monogeneric with just Bogoriella,
Right.
but now also including the genus Georginia.
Gotcha.
And of course,
Yeah.
there's Brevibacteriaceae with its single genus Brevibacterium.
Butenbergiaceae, Bogorialaceae, and Brevibacteriaceae.
Right,
Quite a mouthful.
they are.
What about the rest of the families in this order?
We also have Cellulomonidaceae,
Okay.
which might have some members that actually belong elsewhere.
Oh.
Syngobacteriaceae seems particularly close
Oh.
based on shared cell wall structures, metaquinones, and fatty acid profiles.
It seems there's always a possibility of a family feud in the bacterial world.
There is.
What other families are vying for attention in Micrococcus?
Let's see.
We have Dermabacteriaceae,
Yeah.
known for its unusual peptidoglycan structure,
Okay.
and Dermacoccaceae,
Uh-huh.
with some internal debate about the placement of Ptytococcus.
All right.
And then there's the intriguing Dermatophyllaceae,
Okay.
with its two members, Dermatophilus and Kiniosphaera,
Okay.
potentially on the verge of a separation. Wow, it sounds like a bacterial reality show in there.
Yeah, it does.
Any more families to cover?
Just a few more.
Okay.
We have Intrasporangiaceae,
Okay.
a large and somewhat messy family
Okay.
with numerous genera,
Yeah.
some potentially belonging to other families.
Right.
Then there's Jonaceaceae, a distinct lineage closely related to actinomycetales,
Got it.
and Microbacteriaceae,
Yeah.
a large family with a diverse cast of characters,
Okay.
including the potentially misplaced Tropharema.
Microbacteriaceae sounds like a whole deep dive in itself.
It could be.
And we still have Promicromonospiraceae and Rarobacteriaceae,
Yes.
possibly linked by their cell wall structures,
Yeah.
fatty acids and metaquinones.
That's right.
And finally, there's Rueniaceae,
Yes.
a small but distinct family with just two genera.
That's right.
And with that, we've covered all the families
We have.
within the fascinating complex and often chaotic order of Micrococcatus.
It is fascinating order.
That was a whirlwind tour of Micrococcalis.
It was.
What else is there to uncover in this deep dive into Actinobacteria?
We still have a few more orders to explore within the class Actinobacteria,
Okay.
and then we'll venture beyond this class to see what other surprises the phylum holds.
Sounds good.
Welcome back to our deep dive into the phylum Actinobacteria.
Oh, good.
Before we ventured into that dramatic world of Micrococcalis,
Uh-huh.
we were talking about an intriguing order,
Yeah?
potentially linked to glycomycetales.
Right.
Do you remember which one I'm referring to?
You're talking about Micromonospiralis, right?
Yes.
The one with that genus Actinocatanistra,
That's it.
which might be a close relative of Glycomycetales.
Exactly.
Like a taxonomic crossover episode.
It is.
So speaking of Micromonospiralis,
Yeah.
this order contains just one family, Micromonospiraceae.
Right.
But don't let its size fool you.
No.
This family has its own set of fascinating relationships
It does.
and classification challenges. So what's the inside scoop on Micromonospiraceae? What makes it so intriguing?
The genus Micromonospora, which gives the family its name, is a bit of a wanderer. Its species are scattered throughout the family tree, often more closely related to members of other genera than to each other.
It seems like a recurring theme in the bacterial world.
It is.
Genera with a bit of an identity crisis.
Right.
Defying our attempts at neat categorization.
Exactly.
But you mentioned that there are other genera within Micromonosporiaceae.
Yes.
That are more well-behaved, right?
Yes.
So even within this well-behaved group, there's still some mingling with Micromonospora.
That's right.
What other genera in this family should we be aware of?
We have Acinoa, which represents a very early branch in the family, and Callalatospora, which is closely related to Micromonospora chelsea, the type species of Micromonospora.
Got it.
Despite having a very different appearance under the microscope.
So appearances can be deceiving in the bacterial world.
They can.
Genes and morphology don't always tell the same story.
That's true.
What else is lurking within Micromonosporiaceae?
We have Catelloglobocyspora and Hamadea, a pair recently discovered genera. And Cotinulioplanes, another early branch with several species.
Got it.
And then there's Cuccioplanes, which forms a distinct group with the genera Chryselnicovia and Pseudosporangium.
It's amazing how much diversity exists even within a single bacterial family.
It is.
What's next on our journey through Actinobacteria?
Let's move on to Propionibacterialis. This order is well-defined, containing two families, Propionibacteriaceae and Nocardioidaceae. However, there's ongoing debate about the placement of a couple of genera within Nocardioidaceae.
The drama.
There is. So the troublemakers are Actinopolymorpha and Cribella. While they're currently classified within Nocardioidaceae, our RNA analysis suggests they might actually belong elsewhere in the order.
Another case of genera potentially outgrowing their assigned family.
Exactly.
What's the evidence for this possible reclassification?
Their rRNA sequences place them as distinct lineages within Propionibacteriales, but outside of Nocardioidaceae. And this finding is pretty consistent across different analyses, which makes it even more intriguing.
But there must be some pushback, right?
There is.
Not everyone agrees with this proposed move.
That's right.
Okay, so what's the argument against it?
Well, chemotaxonomic evidence, which examines aspects like cell wall structure and fatty acids, hasn't provided a clear-cut answer. Some features support their current classification, while others suggest a separation is warranted.
It sounds like a classic taxonomic puzzle.
It is.
Until more evidence emerges, they remain in Nocardioidaceae. But with a big question mark hanging over their heads.
Exactly.
Now, shifting our focus to Propionibacteriaceae.
Yes.
This family has several important genera.
It does.
Including Propionibacterium, which is divided into 5 subclades. 2 of these subclades, often referred to as the classical and dairy Propionibacteria, are distinguished by their cell wall structures.
That's right.
It's incredible how even such tiny structural differences can reveal so much about these organisms' relationships.
It is.
What about the other 3 subclades of Propionibacterium?
The 3rd and 4th subclades consist of the monospecific genera, Brooklannia and Propionimicrobium, respectively. While the 5th subclade includes the genus Tesseracoccus. Outside these subclades, we also have the genus Ludiococcus, a deeper branch within the Propionibacteriaceae family.
So even within a single genus, we see a remarkable level of diversity.
We do.
What about the other family in this order, Nocardioidaceae?
Yes.
What should we know about its members?
Despite the uncertainty surrounding Actinopolymorpha and Cribella, the family Nocardioidaceae itself is well defined. The genus Nocardioides, for example, contains a large cluster of species that are closely related to the type species Nocardioides albus, as well as several smaller, more distantly related groups.
So Nocardioides is a diverse but generally cohesive genus.
Yes.
What about the other members of Nocardioidaceae?
We have Aeromicrobium, a genus known for its unique teichoic acids, which are polymers found in the cell walls of certain bacteria. And then there's Marmaricola, close relative of Nocardioides. And of course, we can't forget the potentially controversial genera, Actinopolymorpha and Cribella, each with their unique characteristics and uncertain positions within the family.
It's fascinating how, even within a well-defined family, there's still room for debate and uncertainty.
There is.
What's next on our Actinobacteria adventure?
Next, we delve into pseudonocardiales.
Okay.
This order has undergone some significant changes recently.
All right, like what?
With the merging of two families.
Oh.
Pseudonocardiaceae and Actinocynemataceae into a single family.
Oh, wow.
Now called Pseudonocardiaceae.
It's like a family reunion.
Yeah.
But instead of everyone going their separate ways at the end, they all decide to live together in one big house.
That's right.
What prompted this merger?
Well, a closer look at the evidence, including our RNA analysis, chemotaxonomic data, and even morphological observations.
Got it.
Suggested that these two families were more closely related than previously thought.
So they were separate families. But it turns out they were just different branches of the same family tree.
Exactly.
And what does this newly unified family look like?
It's quite diverse, with several well-defined subclades. One subclade contains genera like Pseudonocardia, Amycalotopsis, and Saccharopolyspora, while another includes Actimocynema, Lechevalieria, and Saccharothryx.
Gotcha.
These subclades are distinguished by some interesting differences.
What kind of differences set these subclades apart?
One key difference lies in their cell wall sugars. The first subclade I mentioned has arabinose and galactose in its cell walls, while the second subclade contains galactose, mannose, and sometimes rhamnose or ribose.
Gotcha.
These subtle differences in sugar composition can reveal a lot about evolutionary relationships.
It's amazing how these tiny sugar molecules can help us unravel the mysteries of bacterial evolution.
It is.
Are there any other notable genera in this newly expanded family?
There's the genus Thermobispora, which was originally placed in Pseudonocardiaceae, but is now considered in order Incertacetes.
Incertacetes?
Yeah.
Can you remind me what that means?
It means of uncertain placement. Essentially, we're not sure where Thermobispora fits in the grand scheme of things.
So another taxonomic mystery.
Yeah.
I love how even with all our scientific advances, there are still so many unanswered questions in the bacterial world.
That's true.
What's next on our agenda?
Let's talk about Streptomycetes. An order with just one family, Streptomycetaceae. This family is home to the genus Streptomyces.
Streptomyces.
Yes.
That name rings a bell.
Yeah.
Isn't this genus famous for producing a vast array of antibiotics?
You're absolutely right.
So over 550 species?
That's right.
That's incredible.
It is.
But you said there's only one family in this entire order.
That's right.
It seems like there should be more to accommodate such a diverse genus.
So there is some debate about whether the genera Chaetocytospora and Streptocediphilus, currently classified as Incertacetes, should be considered distinct from Streptomyces.
So it's like a massive family reunion where everyone's crowding around the Streptomyces table.
That's a good way to put it.
Arguing about who truly belongs there.
Yeah.
What's the current thinking on this?
Well, some researchers believe that Chaetocytospora and Streptocediphilus might just be subgroups within the already massive genus Streptomyces. Others argue they deserve their own separate classifications.
It sounds like a real challenge.
It is.
So Streptomyces is like the celebrity of the bacterial world. Famous, well-studied, but with a complicated family history.
Exactly.
And it's a great reminder that bacterial taxonomy is always a work in progress.
It is.
What's our final order within the class, Actinobacteria?
We'll end our exploration of this class with Streptosporangialis. This order contains 3 families, Streptosporangiaceae, Nocardiopsaceae, and Thermomonasperaceae.
Wow.
They all form distinct branches on the phylogenetic tree. But there are a few interesting twists within this order as well.
I'm sensing a pattern here.
Yeah.
Distinct families, but with internal drama.
That's right.
What's happening in Streptosporangialis that makes it so unique?
Well, the genus Thermobispora, which we discussed earlier as being of uncertain placement, actually pops up here again. It branches off from Streptosporangiaceae, but is currently classified as an order Incertacetus.
So Thermobispora is like that guest who keeps showing up at different parties, but never seems to quite fit in.
Exactly.
So within Streptosporangiaceae, we have several genera worth noting.
We do.
Streptosporangium, for example, is divided into several groups based on rRNA and phenotypic characteristics.
That's right.
Planobispora and Planomonaspora are also notable members of this family.
They are.
Streptosporangiaceae certainly seems like a diverse family.
It is.
With internal groupings.
Yeah.
And an ambiguous relationship with Thermobispora.
That's right.
What about the other families in Streptosporangiales?
Okay.
What can you tell me about Nocardiopsaceae?
Nocardiopsaceae is another family.
Okay.
With some interesting characters.
Like what?
The genus Nocardiopsis, for instance, has a wide distribution throughout the family, with some species potentially warranting reclassification.
Okay.
It's closely related to the monospecific genus Haloactinospora.
Monospecific?
Yeah.
I'm not familiar with that term.
It means that the genus contains only one known species.
Oh, okay.
In this case, Haloactinospora alba is the sole member of its genus.
Got it.
Other notable genera in Nocardiopsaceae include Marinactinospora, also a monospecific genus, and Thermobifida, known for its ability to break down cellulose, a complex carbohydrate found in plant cell walls.
So Nocardiopsaceae has a mix of diverse and specialized members.
That's right.
What about the final family in Streptosporangiales, Thermomenosaurus? What makes this family stand out?
This family also has its share of intriguing relationships. The type species, Thermomenospora curvata, is positioned at the base of the family tree. But the other species, Thermomenospora chromogena, is actually more closely related to Thermobispora.
Wait, so Thermomenospora chromogena might be in the wrong family.
That's a possibility.
And didn't we just talk about Thermobispora being potentially misplaced as well?
Yes, we did.
It seems like there's a lot of uncertainty surrounding these genera.
There is.
Thermomenospora chromogena and Thermobispora even form a distinct branch together in RNA analyses.
That's right.
It's a great example of how horizontal gene transfer can complicate bacterial taxonomy.
It is.
Horizontal gene transfer.
Yeah.
Could you explain that concept a bit further?
So horizontal gene transfer refers to the movement of genetic material between organisms. And it can sometimes make it difficult to trace their evolutionary history. It's like trying to solve a puzzle where some of the pieces have been switched between different boxes.
That's a great analogy.
Yeah.
So Thermomenosporaceae, a family with potential for reclassification. Another example of how horizontal gene transfer can muddy the waters of bacterial relationships.
Exactly.
I think we've thoroughly explored the diverse orders within the class. Actinobacteria.
We have.
What's next on our journey through this fascinating phylum?
Now we'll take a step back and briefly touch upon the other classes within the phylum.
All right.
Remember those classes we mentioned earlier?
Yeah.
Acidomicrobia, Choriobacteria, Nitroliruptoria, Rubrobacteria, and Thermoliophilia.
Right, the classes we briefly touched on in the beginning.
Yes.
I'm ready for a quick tour of these less familiar classes.
Okay, let's start with Acidomicrobia. This class contains just one order, Acidomicrobiales, with two families, Acidomicrobioceae and Iamiames.
Got it.
As their name suggests.
Yeah.
Many members of this class thrive in acidic environments.
Acid-loving bacteria.
That's right.
What kind of environments are we talking about?
Think acidic mine drainage or sulfur-rich hot springs. These bacteria are often involved in the cycling of iron and sulfur in these extreme ecosystems. But they're not typically associated with human infections.
So not likely to cause infections.
Not usually.
But crucial for maintaining these unique and often harsh environments.
Exactly.
What about Choriobacteria?
Okay.
Are they equally extreme?
Choriobacteria is actually quite different.
Okay, in what way?
This class also contains just one order, Choriobacteriales, and one family, Choriobacteriaceae.
Okay.
Members of this class are often found in the gut microbiome.
Back to gut health.
Yeah.
Always a fascinating topic.
It is.
Are these bacteria beneficial or harmful to their hosts?
It's a mixed bag, actually.
Oh, how so?
Some Choriobacteria are beneficial, contributing to the breakdown of complex carbohydrates and the production of essential vitamins. However, others have been linked to inflammatory bowel disease and other gut disorders.
So more research is needed to fully understand their role in the gut microbiome.
Absolutely.
What about the remaining classes, Nitroliruptoria, Rubrobacteria, and Thermoliophilia? Do they also have connections to human health?
These classes are even less well studied. And they're not typically associated with human health. However, they each have their unique characteristics and play important roles in various ecosystems.
So tell me about Nitroliruptoria.
Nitroliruptoria contains bacteria that can break down Nitriles.
Nitriles.
Yeah.
Could you elaborate on what those are?
Sure.
And where they're found?
Nitriles are organic compounds
Okay.
containing a specific chemical structure
Right.
known as a cyano group.
Okay.
They're often found in various industrial waste products.
So these bacteria have the potential to be useful in bioremediation.
They do.
Which is the use of living organisms to clean up pollution.
That's right.
So potential eco-warriors.
Yeah, exactly.
What about rubrobacteria?
So this class contains bacteria with a reddish pigmentation due to the presence of carotenoids.
Interesting.
Which are pigments that can act as antioxidants.
Okay.
They're often found in extreme environments like hot springs and hypersaline lakes.
Red bacteria in extreme environments.
That's right.
That definitely sounds intriguing.
It is.
What about our final class, thermophilia?
As their name suggests, thermophilia contains thermophilic bacteria.
Okay.
They thrive in high-temperature environments like hot springs, geothermal vents, and even compost heaps, where they play important roles in decomposition and nutrient cycling.
It's amazing to think about the sheer diversity of bacteria on our planet.
It is.
From those that live in the human gut to those that thrive in the most extreme environments.
That's right.
And we've only just begun to understand the complexity and importance of these microscopic organisms.
Absolutely.
This deep dive has definitely been a wild ride through the ever-changing world of actinobacteria.
It has.
And it's incredible to think that there's still so much we don't know.
There is.
You mentioned the limitations of using RNA analysis as the sole basis for classification.
Right.
We've covered so much in this deep dive.
We have.
Exploring the intricate world of actinobacteria.
It's been quite a journey.
It's amazing how much diversity exists within this phylum.
Right.
And how much we still have to learn about these microscopic marvels.
There's always something new to discover.
You mentioned earlier the limitations of using RNA analysis.
Yeah.
As the sole basis for classification.
It definitely has its limitations.
It's like trying to judge a book by its cover.
Exactly.
Or a bacterium by a single gene.
Right.
So as we wrap up this deep dive, I'd love to explore the future of bacterial taxonomy.
Sure.
What are the implications of relying solely on RNA analysis?
As we've seen throughout this episode, rRNA analysis, while a valuable tool, can sometimes lead to misleading conclusions or unresolved relationships.
It's like trying to piece together a puzzle with only a fraction of the pieces.
A very good analogy.
So what's the solution?
What?
How can we get a more complete and accurate picture of bacterial relationships?
A multi-gene approach holds the key.
Okay.
By comparing sequences from multiple genes, we can gain a more robust and accurate understanding of how bacteria are related.
It's like adding more layers of detail to that puzzle.
Exactly.
Revealing a clearer and more complete image.
Right.
It makes sense that considering multiple perspectives would give us a more nuanced understanding.
Yeah.
So what specific genes are we talking about here?
So there are several essential genes, often called housekeeping genes, that are commonly used in multi-gene analyses.
Okay.
These genes are involved in basic cellular functions, and they tend to evolve more slowly than other genes.
Okay.
This makes them useful for tracing evolutionary relationships over long periods.
Could you give me some examples of these housekeeping genes?
Certainly, genes like RISA, involved in DNA repair, and JARB, responsible for DNA replication, are good examples.
Got it.
By analyzing these genes, along with rRNA, we can build a more comprehensive picture of bacterial relationships.
It sounds like we're moving towards a more holistic approach.
We are.
To understanding these microorganisms.
Yeah.
And you mentioned earlier that we're entering the age of genomics.
Right.
How does this new era impact bacterial taxonomy?
Genomics is revolutionizing the way we study and classify bacteria with the ability to sequence entire bacterial genomes.
Right.
We now have access to an unprecedented amount of data.
It's like going from a simple black and white photo to a high-definition full-color image.
Exactly.
That's a remarkable leap forward.
It is.
But more data doesn't always make things easier, right?
That's true.
So what does this mean for the future of classifying bacteria, especially those in the phylum Actinobacteria?
It means we need to develop new tools and methods that can handle the massive amounts of data generated by genomics. Bioinformatics, the use of computer science to analyze biological data is becoming increasingly important in bacterial taxonomy.
So what are some of the most exciting advancements in bioinformatics that you're seeing right now?
One exciting area is the development of machine learning algorithms that can analyze vast amounts of genomic data and identify patterns that would be impossible for humans to detect.
So these algorithms can help us to refine existing classifications.
They can.
Identify new bacterial species and even predict the functions of unknown genes.
Exactly.
It sounds like the future of bacterial taxonomy is incredibly promising, but also quite complex.
It is.
So as we wrap up this deep dive, what are your final thoughts for our listeners?
Sure.
Especially those working in the field of medical microbiology.
I think it's important to remember that bacterial taxonomy is a constantly evolving field.
Yeah.
What we consider to be true today might be revised tomorrow. As new data emerges and new technologies are developed,
absolutely,
the key is to stay curious, keep learning, and be open to new ideas.
This deep dive into Actinobacteria has been an incredible journey.
It has.
And I feel like I've gained a whole new appreciation for these fascinating microorganisms.
I'm glad to hear that.
Hopefully this deep dive has equipped you with a better understanding.
I hope so.
Of the key players in the Phylum Actinobacteria, their intricate relationships.
Right.
And the challenges of classifying these incredibly diverse organisms.
Absolutely.
And for our listeners who are eager to learn more, we'll be sure to include links to some additional resources in the show notes.
Sounds good.
But before we go, I want to leave everyone with one final thought. As you encounter Actinobacteria in your work, or even in your everyday life, remember that there's always more to learn.
That's true.
Stay curious, keep exploring, and never stop asking questions.
Thank you for joining us on this deep dive into the world of Actinobacteria